Welcome to the deep dive. I want you to try to imagine understanding how a massive multinational corporation works.
Right.
But instead of reading its corporate rule book, you decide to run an experiment.
Oh, I like where this is going.
Yeah, you just randomly fire, say, 2,000 middle managers simultaneously across the globe. Then you sit back and watch.
Sounds completely chaotic.
Exactly. You watch to see which departments completely collapse, which ones, barely notice, and which ones just go into absolute panic mode.
It's a great analogy, actually.
Well, our mission today is to unpack a monumental 2026 study by Shi and colleagues that basically did exactly that. But instead of a corporation, they ran this experiment on the living, firing mammalian brain.
Which is, it is easily one of the most ambitious biological mapping projects we've ever seen. The scale of the source material we are looking at today is just staggering.
Right, the numbers are huge.
They are. They targeted 1,947 specific genes associated with human diseases, systematically knocked them out across the entire brain of a mouse. And then they analyzed 7.7 million individual cells just to record exactly what happened to the neighborhood.
Which is a completely wild shift in how we actually do biology. Because normally, if you want to know what a disease gene does, you extract a cell, right? You put it in a petri dish and use CRISPR to cut the gene out in isolation.
Right, in vitro.
Yeah, in vitro.
But here's the fundamental problem with that approach. Knowledge is most valuable when understood in context.
Meaning?
A brain is not a petri dish. It's a highly interconnected, incredibly complex neighborhood. Taking a gene out of a cell that's sitting all alone in a dish,
It doesn't really tell you much.
it tells you absolutely nothing about how that cell behaves when it's wired into a living, breathing neural circuit. The cell doesn't have its neighbors to support it or, to react to its failure.
Okay, let's unpack this. Because running this kind of massive genetic sabotage in a living brain in vivo requires some serious sci-fi level technology.
It really does.
If you want to cut nearly 2,000 different genes in a living animal, how do you even deliver that payload without just destroying the brain or creating total noise?
Well, you have to get incredibly creative with a technique called in vivo Perturb-seq.
Perturb-seq.
Got it. Right. And you can't just inject CRISPR scissors and the targeting instructions at the same time. It's too much biological cargo.
The virus would basically be too full.
Exactly. So the researchers started with young mice at postnatal day 16, or P16.
And why that specific age?
That age is critical because their neural circuits are actively maturing. And here's the trick. These weren't regular mice. They were genetically engineered from birth to already express the Cas9 protein in every single cell.
Oh, I see. So Cas9, which for you listening, those are the molecular scissors of the CRISPR system, they are already sitting quietly inside the cells, just waiting for instruction.
You nailed it. Then they performed a retroorbital injection.
But sounds intense.
It's an injection right behind the eye. They inject a massive library of AAV's adeno-associated viruses into the bloodstream there, which gives it direct access to the brain.
And just to clarify for the listener, an AAV is essentially a hollowed-out, harmless viral delivery truck.
Yes, perfectly harmless.
It infects the cell, but it doesn't make you sick. Instead, it drops off a payload, and in this case, the payload is a guide RNA.
Exactly.
It's like a GPS coordinate that tells those Cas9 scissors exactly which of those 1,947 genes to hunt down and snip.
That is a perfect way to visualize it.
Yeah.
So they deliver the GPS coordinates, they wait 3 to 4 weeks for the scissors to make the cuts and for the cells to react.
Right.
And then they extract the brains, and this is where the heavy lifting happens. They use a technology called snRNA-seq.
Single nucleus RNA sequencing.
Right. Specifically a platform called 10x Genomics Flex Apex.
I read about this platform, and the mechanics of it are fascinating. It doesn't just look at the cell, does it?
No, it uses specialized molecular barcodes.
Think of it like assigning a microscopic tracking number to every single piece of RNA inside a cell's nucleus. So when they break the brain down to sequence it, the computer can look at millions of pieces of genetic debris
And reassemble exactly which cell they came from.
and, crucially, which specific CRISPR guide was inside that exact cell. They read both the cellular reaction and the culprit at the exact same time.
Yes, and what's fascinating here is how they organized this massive sea of data. They categorized the brain into 29 different major departments.
Going back to our corporate analogy.
Exactly. These are the 29 binned neuronal types. For instance, you have Peli and Blet, which are the neurons handling excitatory signals in the cortex. or sympaline GABA, handling inhibitory signals.
So they can see exactly which department fired which manager.
Precisely.
But let's talk about the casualties, the dropout effect. I would assume that if you send molecular scissors into a cell to cut out a crucial gene, you'd see a 100% drop in that gene's expression.
Right, you think it's just gone.
Yeah, it's gone. But the data showed an average downregulation of only 18.1%. Wait, 18%? If you physically cut the gene out, why isn't the expression dropping to 0?
Well, you'd think it would be 0. But you're dealing with a living organism's defense mechanisms.
Okay.
When CRISPR cuts a gene, it usually creates a frameshift mutation. The DNA repairs itself poorly, and when the cell tries to read that gene to make an RNA transcript, it's garbled nonsense.
Like a typo in the instruction manual.
Exactly. Now, normally, the cell has a skull check system called nonsense-mediated decay, or NMD.
NMD.
The NMD system acts like a microscopic paper shredder. It recognizes the premature stop codon in the garbage RNA and destroys it before it can make a broken protein.
So the 18% is just what the shredder successfully destroyed?
Right. But biology is messy. A lot of those mutant transcripts actually escape the shredder.
Really?
Yeah. The sequencing machine still reads them floating around in the nucleus, even though they're completely broken and will never, ever produce a functional protein.
Oh, wow. So that 18.1% drop is actually a massive underestimate.
Huge underestimate. The functional knockout, the loss of the actual working protein, is much, much higher.
And the real insight is who survives that functional loss, right? Because it turns out cellular survival is incredibly local.
Highly local.
Like if you knock out a gene like DDX39B, which is an RNA helicase that literally unzips RNA so it can be exported.
Or a gene like HBA5, a molecular chaperone that folds proteins into their correct 3D shapes.
Right. If you knock those out, you get a global collapse.
Because those are your housekeeping genes. They are the building's electricity and plumbing. If a cell can't fold proteins, it dies, regardless of whether it's in the cortex or the brainstem.
It's just a fundamental need.
Exactly. But then you have neighborhood-specific vulnerabilities. Take the gene at TP6V1B2, which controls vesicular acidification.
Basically preparing the shipping boxes the cell uses to send signals.
Right. Or POFA1B1, which handles lipid transport. If you knock those out in the cortex, those neurons are surprisingly resilient. They find a workaround.
But if you knock them out in the midbrain or the thalamus?
Total disaster. Those glutamatergic neurons are highly susceptible. They just die off.
It proves that just because a gene is turned on in a cell, it doesn't mean it's structurally essential for that specific cell's survival. Different neighborhoods have totally different metabolic demands.
Exactly.
Okay, let's lock in what we've discovered so far. Here is your mini recap. First, we've seen how injecting CRISPR directly into a living brain allows researchers to knock out nearly 2,000 disease-linked genes at once.
A huge leap forward.
Second, by applying molecular barcodes to 7.7 million individual cells, we can see exactly who survives the cut. And third, we learn that cellular survival is entirely local. Some genes are global utilities, while others are strictly neighborhood-specific.
Spot on.
Which makes me wonder, and I want you, the listener, to think about this. Look at your own daily workflow. What is the one core housekeeping tool you use that, if removed, would crash your entire day, versus a highly specialized tool that only one specific project relies on?
It really puts cellular dependency into perspective, doesn't it? But this raises an important question. What happens to the millions of cells that do not die?
Right. If the department doesn't collapse when you fire the manager, what kind of chaos is happening inside the office to keep the lights on?
Well, this is where we look at DEGs, or differentially expressed genes.
Okay.
When a cell loses a functional gene, it doesn't just sit there passively. It violently alters its genetic expression to compensate for the loss. Or, it goes into a state of cellular panic.
Panic mode.
Exactly. The researchers found that across all the surviving cells, 27.9% of these reactive DEGs were involved in protein homeostasis.
That's the cell desperately trying to manage a sudden buildup of misfolded proteins, right?
Yes. And another 24% were involved in RNA splicing.
But wait, with nearly 2,000 genes knocked out and millions of cells reacting, isn't that just an incomprehensible wall of statistical noise? How do you even begin to map that panic?
You use a statistical measurement called E-distance, or energy distance.
E-distance.
It's a way to measure exactly how far apart two different transcriptomic states are in a multidimensional space.
Multidimensional space sounds a bit abstract.
It does.
Think of E-distance like mapping the choreography of a panic attack. If two different cells lose totally different genes, but both of them immediately start rationing resources, halting production and stockpiling waste in the exact same sequence.
E-distance mathematically proves they're dancing to the exact same stress routine.
I love that.
And what the E-distance mapping revealed is that perturbations heavily cluster into highly coordinated modules.
Can you give an example?
Sure. The cell has a waste disposal system called the proteasome. If you perturb different components of that system, say targeting a gene like PSMC1 in one cell and PSM4 in another, you don't get random noise.
They react the same way.
Both cells trigger a highly coordinated identical stress response. They share 174 exact same DEGs.
Wow, 174.
The cell detects the trash building up, immediately shuts down normal operations, and activates the unfolded protein response.
But using our corporate analogy again, departments aren't just isolated panic rooms, they rely on each other.
Right.
If the shipping department, which is the cell's vesicle trafficking, starts slowing down, the manufacturing department, the spliceosomes making the RNA, can't just keep operating at normal capacity. They have to form antagonistic relationships.
They absolutely do. And this study mapped those biological tugs of war beautifully.
It's like a literal tug of war.
Yeah. The most striking example involves chromatin remodeling regulators.
Okay, chromatin.
Chromatin is the complex that tightly winds up your DNA. How tightly it's wound determines whether a gene can be read or not.
Got it.
If you knock out genes like Y11 or BRD4, which are normally responsible for unwinding DNA and activating transcription, you push the cell into a very specific lockdown regulatory state.
But if you knock out repressive chromatin regulators, the ones responsible for winding the DNA shut, the cell is pulled in the exact opposite transcriptomic direction.
Exactly. Everything gets less wide open. It's a literal molecular tug of war keeping the cell balanced. The resilience of the cell relies on this constant opposing tension.
So if you cut the rope on one side, the other side goes flying.
Exactly.
Let's pause and synthesize this. Time for another mini recap. First, we know that surviving cells dramatically change their gene expression, creating DEGs to compensate for lost genes.
Right.
Second, thanks to E-distance mapping, we can see that disrupting different genes within the same complex like the waste disposal system triggers an identical choreographed cellular panic. And third, we learned that biological systems are fiercely antagonistic, constantly pulling the cell's state in opposite directions to maintain a delicate balance.
Perfectly summarized.
So, listener, ponder this. If we think of cells as trying to maintain equilibrium through tension, where in your life do you see two opposing systems keeping things stable, where if one fails, the other immediately goes into overdrive?
That concept of opposing forces is so critical here because it perfectly sets up the most mind-bending clinical finding of the entire deep dive.
Yes. Here's where it gets really interesting. Let's pivot from these massive global brain networks to a highly specific clinical mystery that has stumped neurologists for years.
Oh, the NMDA receptor.
Exactly. It involves a massive protein structure called the NMDA receptor.
So the NMDA receptor is a crucial intersection in your neurons that binds to the neurotransmitter glutamate. It is the absolute bedrock of memory, learning, and synaptic plasticity.
It's essential.
Absolutely. Now, there are two genes, Grin2a and Grin2b, that encode subunits for this exact same receptor. On paper, they make parts of the same machine.
But clinically, they are totally different beasts. Mutations in Grin2a are heavily linked to schizophrenia.
Right.
But mutations in Grin2b are linked to autism spectrum disorder and severe neurodevelopmental delays. If they're just two subparts of the exact same receptor, why on earth do they cause entirely different human diseases?
Well, for a long time, the clinical dogma was simply about timing. Grin2b is highly active early in fetal development, while Grin2a comes online and matures much later, after you're born.
So they just assumed it was a timing issue.
Yeah, the field assumed the different diseases were just a result of the brain being disrupted at different stages of its construction.
But this study proves that assumption is fundamentally missing the biggest piece of the puzzle. Because of the sheer scale of the Perturb-seq data, the researchers didn't have to guess anymore.
They could just look.
Right. They could look at the exact same cell type, specifically layer 23 IT cortical glutamatergic neurons.
These are the excitatory neurons sitting right in the upper layers of your cortex.
They knocked out Grin2a in some of these cells and Grin2b in others at the exact same point in time.
And what they found completely rewrites how we view these genes. The transcriptional responses in that exact same cell type were strongly anti-correlated.
How anti-correlated?
The Pearson R correlation was -0.516.
Let's translate that statistic, because a negative 0.516 isn't just a minor difference. It means these genes aren't just doing different things, they are actively running in opposite directions. For every step the cell takes to the left when Grin2a is missing, it takes a step to the right when Grin2b is missing. It's a completely divergent genetic algorithm.
Think of the cell like a commercial airliner. And Grin2a and Grin2b are two co-pilots sitting side by side in the cockpit.
Okay, I like this.
For years, we thought, well, if you remove either co-pilot, the plane just gets a bit wobbly.
Great.
But what the transcriptomic data actually shows is that if you remove pilot A Grin2a, the plane suddenly pitches sharply upward.
And there's a cellular logic to this panic, isn't there? When pilot A is lost, the cell desperately upregulates genes tied to activity-dependent plasticity and presynaptic release, genes like BDNF and NPTX2.
Yes, it thinks it's losing its ability to communicate.
So it wildly ramps up its neurotransmitter release to just shout louder into the void.
But if you remove pilot B Grin2b, the plane pitches sharply downward. The cell does the exact opposite.
So it stops shouting.
It downregulates those communication genes and instead wildly upregulates structural remodeling genes like TMAN1. The cell thinks its actual physical structure is failing.
Oh, wow.
So it ignores communication entirely and pours all its energy into reinforcing its physical membrane.
It's a completely different survival strategy based on which nearly identical part you removed.
And this brings us back to our core belief today. Context is everything.
Right.
You could never see this dynamic in a wild-type tissue sample, and you could certainly never see it in a petri dish. You have to observe the cell desperately trying to survive within its living firing circuit to realize that these two identical parts are hooked up to completely different emergency alarm systems.
That is just incredible. Let's process that before we move on. Mini recap time. First, Grin2a and Grin2b make parts of the same receptor but cause wildly different brain diseases.
Yes.
Second, we used to think the difference was purely about the timing of when the genes were active during childhood. But third, we now know that in the exact same living cell, losing one gene triggers the exact opposite genetic survival reaction as losing the other.
Opposing chain reactions.
So, question for you, the listener. Can you think of two things that seem nearly identical on the surface, like two software updates or two identical car parts, but they cause completely opposite chain reactions when they break?
It completely reframes how we think about targeted therapies, which brings us to the ultimate challenge here.
Right, the scalability nightmare.
Exactly. So if Grin2a and Grin2b are just two genes and they cause completely opposite molecular panic attacks,
multiply that by the thousands of disease genes in the human brain. Does that mean we need to invent thousands of completely different, highly customized drugs? Are we looking at a biological scalability nightmare?
that is the trillion-dollar question. Because the data showed that neurodevelopmental and autism-linked genes do not disrupt the brain uniformly.
They act like clinical snipers.
They really do.
Yeah.
For example, if you perturb the CystXBP1 gene, it massively disrupts cortical layer 4 and 5 neurons. Specifically, it drives down a synaptic gene called NRXN2.
But if you look at another one,
Right. Meanwhile, perturbing CTNB1, which is a totally different autism risk gene, wreaks havoc entirely outside the cortex. It heavily impacts the thalamus.
so it feels incredibly fragmented. If every mutation breaks a different cell in a different way, how do we ever cure these diseases?
Well, here's the most hopeful finding of the entire deep dive.
Yeah.
The researchers took the top 5 targets with the strongest neurodevelopmental disease enrichment.
These are heavily studied genes with names like SLC17A5 and anchored 11.
Right. They mapped every single DEG, every downstream ripple those mutations cause, and threw them into a massive protein interaction model called a string network.
And a string network is basically playing 6 degrees of Kevin Bacon, but for proteins.
I love that, yes.
It maps how a disruption in one corner of the cell eventually bumps into proteins in another corner. And despite SLC17A5 and anchored 11 being completely different genes, causing damage in completely different ways, their downstream ripples eventually crash into the exact same wall.
Yes. They heavily converged on a single module, the NMDA receptor and postsynaptic signaling. If we connect this to the bigger picture, think about a major city. You can have a traffic jam caused by 50 different things.
A flat tire in the north, construction in the east, a parade in the west.
Exactly. But eventually, all that backed up traffic funnels down, and completely paralyzes the exact same downtown intersection.
So the clinical algorithm completely changes.
It does.
If 50 different rare genetic mutations all eventually cause a downstream traffic jam at the exact same molecular intersection, you don't need to invent 50 different drugs to fix the 50 different flat tires.
No, you don't.
You just need one drug to act as a traffic cop and clear that specific downtown intersection.
And that is the promise of genome-informed therapeutic design. We are moving away from just trying to replace a broken gene. Instead, we map the entire cellular neighborhood's reaction to that broken part. We find where the panic converges, and we soothe the neighborhood.
All right, time for our final recap to bring it all home. First, we've learned that disease genes impact very specific, distinct cell types, acting like snipers rather than uniformly damaging the whole brain. Second, despite this specificity, different genetic mutations often funnel their downstream damage into the exact same shared molecular pathways. And third, this convergence offers a roadmap for therapeutics, allowing us to target the shared traffic jam rather than the individual genetic cause.
Such an elegant way to solve it.
So, listener, look at complex problems in your own industry or life. Are there multiple different errors or crises that all ultimately bottleneck at the exact same shared node?
It's a powerful framework for problem solving well beyond biology.
So what does this all mean? We started by talking about how 7.7 million cells were mapped. Not just to see what genes are, but to prove what they actually do when you remove them from a living system.
Yeah.
We've gone from looking at static, muddy diagnostic X-rays to watching the living, breathing traffic of the brain in real time.
And we've learned that biology is remarkably modular. The brain's immense resilience relies entirely on these delicate antagonistic tugs of war. But there is a vital caveat to this research. This study looked at loss-of-function mutations at a single snapshot in time in very young mice.
And that leaves us with a fascinating and maybe slightly provocative final thought for you to mull over. This study proved that our brain's resilience is based on these delicate, opposing forces constantly adjusting to damage.
Right.
But what happens to those ropes as we get older? Imagine if we could map the brain across an entire lifespan, watching how these genetic traffic jams evolve as we age.
It would be incredible.
Think about how your own neural networks right now might be actively compensating for genetic quirks you don't even know you have, masking a molecular tug of war that's been happening in your cortex since the day you were born. The diagnostic waters used to be muddy, but the sheer scale of data is finally starting to clear them.
It truly is a new frontier for genetic medicine.
Thank you so much for joining us on this deep dive. Keep questioning the obvious, and we'll catch you next time.